Okay, so today's class is on the various methods of sequencing for a particular DNA stretch. Obviously, not limited to gene. Any stretch of DNA, be it non-coding or a coding gene, whatever, if we can sequence it, and which are the most common methods. So, in this context, I'll be discussing with you Sanger sequencing and then automated sequencing and one another technique called pyro sequencing and then a version of pyro sequencing that I'll be talking. So directly coming to the Sanger sequencing method. So let's say you have to sequence this particular stretch of DNA. Okay. So, how will you do that? Because obviously, in molecular biology, you can't see things that, okay, now DATP, A has been added by DNA polymerase, now C has been added, and so on. So, obviously, you have to figure out a method to detect that which all nucleotides are there and in which order they are there. So let's say my ultimate aim in this particular sequencing method, which is called Sanger, because it was Dr. Sanger who developed this technique. So in this, my aim for now is that I want to get the fragments, each differing by single base. Okay. So what I do is that in my reaction mixture, I do have this single-stranded DNA, and then I add primer to it, which is complementary to certain bases towards one end of the DNA. And I add DATP, DCTP, DTTP, and DGTP, means all the four nucleotides for the reaction to take place, and obviously DNA polymerase will be there. So if I add all of these things, so it is a typical polymerization reaction, and as and when a particular nucleotide comes, which is complementary to the nucleotide which is present in the template strand, it will be added. But what I do is that apart from these four nucleotides, which I just mentioned, what I do is that I divide my reaction mixture in four different tubes, and in each tube I add one extra nucleotide, which is dideoxynucleotide. So in one tube, apart from everything that I mentioned, means template, primer, all four nucleotides and DNA polymerase, I add DDATP, dideoxy ATP. So in this, how does this particular nucleotide differ from the general nucleotide is by the fact that apart from the absence of the 2' OH group, these nucleotides also lack the 3' OH group. And what will be the consequences of the absence of 3' OH group will be that no further phosphodiester bonds will be made. So let's say if in your reaction a DDNTP gets incorporated, so the chain will be terminated there and then. Why it will be terminated? Because it doesn't have the OH group here, it just has the H group here. And so the phosphodiester bond for the incoming nucleotide cannot be made. Okay, so one point is this. So in one tube, I have, let's say, added DDATP. So in this particular reaction, which is happening in a tube, I will have different size of the PCR products, all of which are ending in DDATP, I mean A, okay, which is complementary to T. So means that I will have one fragment which will till here, I will have one fragment which will end here, and I will have one fragment which will end here. And I also should mention that the DD NTPs that we add, they are radioactively labeled. Okay. Then in another tube, what I do is that apart from everything which is present in the reaction mixture, I add DD CTP, let's say. So it means that in that particular tube, I will have PCR products of varying length, all of which will be ending in DD. What did I say? That what we are adding DDGTP, let's say. So all of these will be ending in G, and those G will be complementary to a position where C was present in the original template strand. So it means that in different tubes, I will have different fragment lengths, all of which are adding in a particular nucleotide. So then what I do is that the products from 4 different tubes, I run it on sequencing gel. And so this is the tube in which I had added DD-ATP, and in this tube I had added DD-CTP, and so on. Okay. So now when I run it on gel, I can say that, okay, this particular, and because I told you that these nucleotide, the DD-ATP, CTP, the dideoxy ones, they are all radioactively labeled. So now I can deduce my original sequence of the template DNA by this method. So I would know that, okay, this is the shortest fragment. And so it means it is most close to the primer which I had added. Okay, so obviously something which is of this size is the one which is most close to where the primer was complementary. So it means the sequence would be G, then A, then C, then T, then G, and A, and so on. Okay. And so this is the sequence of the complementary strand, because I know the identity of the nucleotides at which this particular fragment is ending. But that base is complementary to my original DNA stretch for which I wanted to know the sequence. So then how I can deduce the complementary strand is just by having a complementary DNA. So then it means that my original DNA had this sequence A, G, C, T, T, and so on. Okay, it is complementary to this. So if in exam you are given a stretch, means this particular gel photo, and you are asked to deduce the sequence of the DNA, then this is how you will be deducing. So I hope that you understood the logic behind it and how it is performed. If it is not clear, please ask me. Are you all listening? Am I audible?
Yes, ma'am.
Okay.
Yes,
So, okay. So is it clear?
Yes, ma'am.
Okay. So this is the Sanger chain termination sequencing method, which was done previously. And as you can see, it is not automated. Here, what we have done is we perform PCR reaction in 4 different tubes. And once the PCR is done, then we run it on gel. And this gel is very big gel. The simple, so it's a vertical gel. It is a polyacrylamide gel. Made with such porosity that even the DNA fragments with one base pair, they can be read easily. And their size is very big. Means for me, loading the sequencing gel was very difficult. During my PhD days, I've run a few sequencing gels. So it was very cumbersome. Also, there was one problem that because these means I told you that these DDNTPs that we are adding, they are radioactively labeled. So obviously, I had to wear these gloves. We had to wear. We had to perform all the reactions in front of a radioactive shield. So everything was difficult. Even when you have to do the centrifugation steps, etc., then it has to be done separately in another centrifuge, which is kept separate from the other centrifuge in the lab, just in case there is some radioactivity spillover. Even you have to be very cautious with the pipettes and all that you have to use. So these are some problems that you encounter when you are working with radioactivity. And also I told you that you had to run it on very big gels and then deduce the whole sequence of a particular DNA stretch. Now, same thing, the same ideology of chain termination using DDNTP means Sanger sequencing. Now it is automated. So now what they have done is that they have taken these 4 fluorescent DD-NTPs. Okay, so instead of radioactively labeled, they are fluorescent, and each DD-NTP has a different fluorescence. So you can differentiate when DD-ATP is there and versus DD-CTP there, because all have 4 different fluorescence. Another thing that they have done is that they all pass it. There are because they all have 4 different fluorescence, so which the signal cannot be mixed. Unlike, let's say, if in this case, when it was radioactively labeled, just imagine if I had pulled down the PCR reaction from 4 different tubes and run it on gel, I would not know that this thing, this radioactive signal, which will appear black in the final, when I finally develop my radioactive film, it comes from which particular DDNTP. So that's why we had to run 4 different gels. But because now we have different fluorescence for each DDNTP, it has become easy. So now what I can do is I can have a thin capillary and then pass all the 4 reactions combined through this capillary. The reaction still would have to take place in 4 different tubes. It's just that after the PCR is done, you combine these and then you run it through this capillary. Now, what will happen that let's say there was a fragment which was ending in this TTP, okay, T. So it means it has a red fluorescence. So when this fragment reaches here and this laser beam means it passes through it, then there is a photo multiplier, and I can finally detect that, okay, this is a fragment. This small fragment is having T at the end. Then the next fragment will come, and then again laser beam will shine, and then I can, okay, deduce that after that there is a, and so on. Okay, so this is that I have reduced my work. One, that there is a safety issue which was there for radioactive, that I've taken care of, and also I don't have to run 4 different gels, mega gels. So there is a thin tube, and as and when the fragment passes through it, we come to know that it's ending in which particular nucleotide. So this is the method that is currently being used, Sanger sequencing. When, for example, you do a cloning and your teacher asks that please send it for sequencing. So what you do is you just send the PCR product or your DNA fragment to the company. The company then performs PCR using this method and finally gives you an output. How does the output look like? It looks like this. Okay, I'll just come to that. But I think that this thing is clear, that as and when these fragments are passing through this capillary, according to their size, you can figure out that these fragments are ending in which particular nucleotide. Okay, if this is clear, then I'll move forward. Okay, so silence is that you understood. Okay. Okay. So, how will your output look like in the this particular, means what you are given by the company is this particular output. Apart from the sequence that has been read, they also give you these peaks. Okay. So these peaks show that, okay, means let's say your C was green in color. So you will see a very clear peak for C here. But for rest of the nucleotides, you will not see any peak. And if next is A, you will see a peak here, and it is through these peaks that you figure out that which nucleotide is there. In some cases, you are seeing that the peak is small, and in some, you are seeing that the peak is long, means it is tall. So, does it have any significance? No, it doesn't have any significance in this particular method of sequencing. Even if the peak is small, I would still say that there is a single C here. Okay, and so on. Only problem would be that if you have overlapping peaks, many times you will find that when you get your sequence back from the company, you will see that there are overlapping peaks. Let's say you have C and G overlap. So in those cases, it means that you are not able to read the sequence properly, and you have to get it resequenced from the company, because then you cannot figure out whether it was C or G. So because of some problem, and sometimes it happens in CG rich region, or in region when there are multiple nucleotides at a stretch, at times you do have problem in figuring out that what's the real sequence. So many times you get both the strands sequence and then come to a conclusion. If they both are giving the same sequence, it means that that sequence is right, or you have to get it resequenced if you are getting overlapping peaks. But I again want to emphasize that the length, this size of the peak is no indication of multiple these. Oops, let me just hide it. Oh, oh, oh. Can you see my, is this visible?
Yes, ma'am.
Acha. So what I
Yes,
want
ma'am.
to say, ठीक है, thank you, thank you. What I want to say that, see, this peak size is big. So it doesn't mean that there are multiple T's here. The two T which are shown here are solely on the basis of two clear peaks of T. Okay, so why I'm emphasizing this? Because in another method, the size of the peak matters, and then you have to deduce that how many nucleotides are there at a stretch. So this is everything about the automated method of Sanger sequencing, and you can understand that it is much easier as compared to the previous methods. So yeah, so this figure also shows the comparison. In this, 4 different lanes were there. Here a single lane is there, and as and when the fragments pass, you can figure out which nucleotide was there in that particular position. Then I want to tell you that which enzymes are used for DNA sequencing, for Sanger sequencing. So as I told you that this relies on polymerization, right? So it means obviously some DNA polymerase has to be there, which will recognize a free 3' OH group, as shown by this primer, which you always use for Sanger sequencing. And once you have a free 3' OH group, then any DNA polymerase can come and do the perform this polymerization reactions. So the enzyme that we use is clinopolymerase. Uh, it used to be clinopolymerase. I will actually take you through a whole journey of the current sequencing enzymes that are being used, and when you will see the innovation, the hard work they had put together to actually arrive at a good sequencing enzyme which doesn't have the problems that are faced during sequencing reaction. You will understand, you will means also connected to some of my previous classes where I told you how do you manipulate DNA, etc. I'll just come to that. But for now, I just want to tell you that there is one enzyme, clinopolymerase. It has low processivity. What is processivity? It means that it can synthesize a very short DNA strand before dissociating from the template. So in this case, we do not want that any error in reading sequencing should happen just because my DNA polymerase was not processive enough, just because it came off the template, and that's why the chain got terminated. It shouldn't be the case. It should only be terminated when a ddntp comes. Okay, so in general we prefer a DNA polymerase which has a very good processivity. So then I just come to all these points in this particular slide. Okay, so this is very beautifully given in this particular article, and actually when I read this article I was quite hooked to it, so I wanted to share this whole journey with you. So when you're looking for a sequencing enzyme, one thing is that you do not want it to be compromised in its processivity. So one point is this: it should be highly processive. Then it should not have exonuclease activity, because let's say when you put a ddntp towards the end of a particular means the chain is ending in a particular DDNTP, which you want to finally monitor to deduce the sequence. But because of, let's say, some 3' to 5' activity, that gets removed. So because of that, there will be error, or I'll not be able to deduce the sequence properly. So generally, we prefer enzymes which don't have much of exonuclease activity. Then the third sequencing enzyme property that we really seek for is the fact that it should not discriminate between DNTPs and DDNTPs, because when we are doing that polymerization reaction, we want that a random DDNTP should also get incorporated at various places apart from the DNTPs that are being added. So there should be no discrimination between DNTP and DDNTP. So with these three properties, let's see how they finally arrived at the current sequencing enzymes that are currently in the market. So the E. coli DNA polymerase 1, it has very low processivity. So then it turned out that they were working on a particular E. coli strain in which E. coli DNA polymerase was mutated. Okay. So it means in that strain, this DNA polymerase is not being made. So instead, they express T7 DNA polymerase and in presence of certain enzyme. So what they found that T7 DNA polymerase, it was definitely functional in E. coli. So they found that the T7 DNA polymerase is also able to do function of polymerizing in E. coli. And second is that when they added thioredoxin to it, which I discussed in my previous class, the processivity of the polymerase increased many fold. And in this article, it was written up to 100-fold processivity increase. So obviously, this is a very big plus point, which was not present in the previous enzyme. So then it was realized that, okay, now we know that if we use T7 DNA polymerase, it will be better as compared to E. coli DNA polymerase. But then what about the exonuclease activity? How do we remove the exonuclease activity in this? So in this, then by performing several experiments, they came to a conclusion that the exonuclease activity could be taken care of if we perform the reaction of polymerization under reducing conditions, a reducing agent and ferrocyanine in presence of these things. What we found that exonuclease activity of T7 DNA polymerase was highly compromised without affecting its polymerization activity. So it means not only we have a highly processive enzyme, but also we have taken care of exonuclease activity. And the third point, the discrimination between DNTP and DDNTP. The problem with the E. coli polymerase was the fact that it was highly discriminating between DNTP and DDNTP. But then it was realized that T7 DNA polymerase did not differ much between these two. So whether DNTP comes or DDNTP come, it was able to do it better, much better as compared to E. coli DNA polymerase. But then, as the molecular biologists do, they try to, improve over the previous versions. They found that in the PCR reaction, when they replaced manganese, means magnesium with manganese, the differentiation between DNTP and DDNTP, it was reduced further. So means more improvement than the previous T7 DNA polymerase. The advantage just came with the fact that you, in the PCR reaction, you replace magnesium with manganese. Then again, the sequenase enzyme was developed. So they were trying to look for the fact that can we have these exonuclease activity, almost completely removed. So in that case, what they did, that they were doing some these mutagenizing screens and chemical screens, etc. And they came across a mutant which lacked the 28 amino acids in the N-terminal region. Okay, so it was some random screen that was happening, and they found that the mutant which lacked this particular domain had no exonuclease activity left. Okay, almost it was nil, but its polymerase reactivity was higher than the previous version. So means if you remember, I told you about the puck vector. It was a chance mutation in the ORI, and if you remember that that chance mutation, it increased its copy number several hundred folds. So in here also, not only it decreased its exonuclease activity, but its polymerase activity was also higher than the previous version. So this sequenase 2 enzyme is being used in many labs for sequencing purposes. Okay. Then came the version that when we are doing PCR, we use the polymerase enzyme, which which means in PCR we know that there are repeated cycles of heating, then cooling, then heating and cooling. So wouldn't it be better than rather than these polymerases, be it T7, be it E. coli, be it Clino, whatever these various polymerases were being used, which are not thermostable, how about we use a thermostable enzyme? Okay. So actually, in a way, I've given notes for you here, but please go through this article. It's written in a very interesting manner. So what they did was that let's try these tag DNA polymerases or various thermostable enzymes, be it Vent polymerase or tag polymerase, etc. So the advantage was that these were thermostable, but the disadvantage was that the DDMTP, they had a very, very strong discrimination against the DDNTP, the property which was not seen in T7 DNA polymerase. So one thing was that, okay, how about you add manganese, which they were initially doing it here. Manganese, yes, it can be added, and definitely it improves the discrimination between means it shows less discrimination between DNTP and DDNTP. But it comes with its own disadvantages, in which it was seen that the working concentration was very narrow. It led to precipitation. And okay, I'll have to move to some other room. There's a lot of disturbance here. Wait. So, am I audible again?
Yes, ma'am.
Okay. So then what they did was that so means one option was that you go for tag DNA polymerase in presence of manganese. Then they did, mean some other labs were doing a lot of molecular biology experiment, and their idea was that let us compare the DNTP and DNA binding, this activity of E. coli DNA polymerase and T7 DNA polymerase, because as a basic molecular biologist, they wanted to understand that what is there in the binding domain of T7 DNA polymerase, that it is not able to discriminate between DNTP and DDNTP, but E. coli DNA polymerase was doing so, right? So what they did was that they started swapping the domains between E. coli and T7 DNA polymerase. And this I will also, briefly mention when I'll be telling you about mutagenesis class, that how do you do domain swapping, etc. But yes, here what they did, they started swapping the domains between these two. And through these domain swapping experiments, they finally came to a region called O region, that okay, it is this O region which is responsible for T7 DNA polymerase to not differentiate between DNTP and ddNTP. Then, like molecular biologists, they want to understand in more detail what is there in this domain that makes it, so efficient in not discriminating, etc. So then they started doing site-directed mutagenesis, and through those site-directed mutagenesis experiments means mutations at specific sites or even random mutagenesis in that particular domain, they finally arrived that it is the tyrosine 526 amino acid in the T7 DNA polymerase, which is responsible, means it was found as the single determinant for discrimination between DD and TP. So, because now we know that this tyrosine is responsible for the good properties that we want in the sequencing enzyme, what they did was that this corresponding residue in tag DNA polymerase, they replaced that with tyrosine. And then they found that now let's see that this tag polymerase, which definitely has very good ability, it's not a, it's a thermostable enzyme and it is a good polymerization enzyme, etc. The only problem was the discrimination. So then will it increase or not? And to surprise, it was beautifully, it showed drastic results. You can read it that the modified tag DNA polymerase F667 to Y, that is when it was converted to tyrosine, it started preferring ddNTP rather twofold over dNTP. Can you imagine? Here it was, the discrimination was the other way around, from 100 to 10,000 fold, or let's say up to 6,000 fold, etc., whatever. But now it started rather preferring other way around, or it was similar. So this is how a thermosequenase was developed by the molecular biologist. So let's say if you are, you join a company after your PhD or whatever masters, and you are given an enzyme to improve upon, these will be the various strategies that you have to use to improve it. So means this, journey, I find that it's very important for all molecular biologists to know. Okay, so now till now what I've told you is the Sanger method, the manual method and the automated method, and then I also introduced you to the various sequencing enzymes that are currently being used for sequencing purposes by Sanger method. Then I think you have been introduced to the term called universal primer. Just wanted to tell you that, let's say you have cloned 10 different fragments in the, let's say, pcDNA3 vector, which is present in your lab. Now you have done PCR and you did the ligation and then you cloned it. Now you want to know whether the fragment that you have cloned, are they really the right sequence or not. So in those cases, you have to go for sequencing. So how will you go for sequencing for 10 different fragments in your lab? Will you design individual primers for each of these and then go for sequencing? No. So in all of these vectors, there is a certain region. Let's say in this, a T7 primer region is given, for against which all the companies would have a primer. So they already have a primer. So what I have to do is I just have to send my 10 recombinant clones to them, and I ask them that please sequence it using T7 primer. Why T7 primer? Because my this commercial vector that I used, it does have a T7 binding site. T7, I mean sequence in there. So this universal primer by the company can be used to sequence all of your 10 different products. So that is a simple advantage of having a universal primer site. So all the companies, they have either for T7, for SP6, for M13. So many different generic primers are there with the various sequencing companies which are present throughout the world. Now comes the problem that what about if your cloned sequence is more than 500 base pair. Let's say it is because with Sanger sequencing you can get, let's say, up to nowadays it has improved, so it is up to like 7 to 800 base pair. You will get good sequencing results for a particular stretch of DNA, but beyond that it is not much efficient, because obviously there is something called as processivity also, and so there are certain limitations, so it cannot go beyond that. So in those cases, what you have to do for bigger inserts, you have to design your primers one by one. So let's say you have, let's say, a 10 kb sequence. So you send to a company and you say, please, using a universal primer, you tell me the sequence of the first 5, 600 or 700 base pair. So once you get the sequence, then you again design the primers based on the sequence you got. You design this primer and then you send to the company, and then you get further 500 or 700 base pair, and so on. So this is this technique I'll also revisit it when I'll be telling you about genome sequencing, because this is called as walking method, that you go step by step. You know the sequence here, then you go further, then you go further, then you go further, because you are limited by the fact you don't know the sequence in between. So once the sequence come, you design the primer and then you sequence it further. Okay. So these are primers, they would be called as internal primers. So this slide is to sensitize you to the two terms, universal primer and internal primer. Now coming to another technique, which is pyro sequencing, which was means better, means less time consuming in terms of as compared to the automated Sanger sequencing also. See, in automated sequencing, what this Sanger sequencing, I would say, what we had to do is we had to do this polymerization uh, with in 4 different tubes using DDATP and so on. And once the PCR is done, then we had to pass it through a capillary. So we'll have to wait that, okay, once the PCR is done, then they'll be passed, and according to their length and the fluorescent signal that they have, we will come to know of the sequence. So we'll have to wait. We are dependent on that. But then the idea came that how about as and when a nucleotide is being attached here, we get to know of the sequence. We get to know of the identity of the nucleotide, which comes and binds to the, makes a phosphodiester bond with the primer, and with the next, and with the next, and with the next. So as and when the nucleotide comes, if we can know its identity, was the idea behind pyro sequencing. Okay. So in this, what they have done is that what they do is, let's say, the first base here in template is C. Okay. So now what I do is I add DATP here. So it will not make a phosphodiester bond here, because it is not complementary to the template base. So what I do is that in my reaction mixture, which will have template DNA and the primer and this sequencing enzyme, means any polymerase, I also add apyrase, or also called as, let's say, a nucleotidase. I mean, any nucleotide which is not being utilized, it should be quickly degraded. Okay. So in my this reaction mixture, I also have a apyrase. So if DATP was not used, it quickly gets degraded. Then I add DTTP. So in this, my reaction mixture does not already have the nucleotides. Here you can see the difference. I'm adding the nucleotides one by one. Okay. Then I add DTTP. Again, this is not complementary to the C which was present there. So it will again get degraded. Now let's say when I add DGTP, what will happen? It will come here complementary to it, and the phosphodiester bond will be made. Because the phosphodiester bond will be made between the previous nucleotide and the incoming G, the this pyrophosphate, it will be released. Okay. Somehow we have to get a method to detect that which nucleotide has been incorporated here. Unfortunately, we cannot look at the nucleotide which has been incorporated. We have to deduce it smartly through certain reactions which give the output in terms of color or fluorescence. So what is happening here that when I know कि मैंने DDTP डाला and pyrophosphate has been released. So how do I detect that pyrophosphate has been released? By a very smart reaction which is given here. So let's say the nucleotide gets added by the polymerase, PPI gets released. Then I know through my knowledge of chemistry and biochemistry, I know that when this in presence of adenosine 5' phosphosulfate APS and in presence of the enzyme called ATP sulfurylase, it releases ATP. Okay. Now, how do I detect that? Okay, ATP has been released. So for that also I have a very smart method. So ATP in presence of luciferin gives this particular reaction, which finally gives a chemiluminescent signal. So it means that this signal can be detected finally. So finally, what I want to say that when I add DDTP, and what will happen? PPI will be released. PPI will be then leading in presence of APS and other things will give ATP. ATP in presence of luciferin, etc., and luciferase enzyme, it will give chemiluminescent signal. So whether my DGTP has been incorporated here, I can easily see with the help of the signal. So as in when I don't have to wait, I am adding the nucleotides, I know the identity. Then what I do, I go sequential. So pehle A, T, G, C, let's say is in the order. So then I add DCTP, it gets degraded. Then I get add DATP, I will see a, let's say, chemiluminescence. So it means that after G, the sequence was A. Okay, so it means that now I can read the sequence as and when it comes, the nucleotide gets incorporated. So because it is like addition of these nucleotides one by one, so obviously this whole thing, it can be automated. So this is now automated, and what we see, the machine, what it does is it adds these various nucleotides one by one, and in the reaction mixture you have this apyrase. You can also think of it as a nucleotidase, which is that the nucleotides which are not getting incorporated, they get degraded immediately. Because let's say if they are not getting degraded, let's say if they stay in the system, then it will lead to a lot of confusion, because we are only relying on the fact that in this step I have only added DCTP. अगर मेरा पहले का DCTP भी already होगा और मैंने DCTP नया नहीं डाला है, then it will give me a false signal, something like that. Okay, so it has to be stepwise, and I have to ensure that the previous nucleotides, they do get degraded. So in my reaction mixture, all these things would be there, and finally my detector, it will give me the signal. So that signal again is in the form of peaks. So how can I read my signals here? So in this case, it means my nucleotide incorporated with, when I added, I get A here, then T, then G did not get incorporated, then C got incorporated, then A got incorporated, but then I got a longer peak. But in here, the longer peak means that A got incorporated again. So the height of the peak will tell me how many A's got incorporated. अगर क्योंकि इस केस में means let's say I added DATP, तो सिग्नल आया, लेकिन इसके बाद भी ATP है तो वो क्विकली इनकॉर्पोरेट हो गया, फिर उसके बाद ATPF तो फिर से इनकॉर्पोरेट हो गया. So it means it's getting incorporated in quick succession, and what I get is a cumulative chemoluminescence peak. So unlike in case of Sanger sequencing, in here it is the height of the peak which will tell me that how many nucleotides are there. Okay. So then T and G did not get incorporated. Then when I added C, then I saw the signal. So then what is my sequence? A, T, C, double A, and C. Okay, so this is how using pyrosequencing you can deduce the this sequence of a particular stretch of DNA, and the advantages of pyrosequencing as compared to Sanger sequencing, even though Sanger was automated, is the fact that you don't have to wait and run it through the gel and then get to know of the identity of your sequence. So is pyrosequencing clear? These are little complicated techniques. So
Yes,
I'm
ma'am.
perfect. Acha, did somebody say no? Okay, so then I'll move to the massively parallel pyro sequencing, and this is a means believe me, when you will see that how this technique is, you will be almost in lot of admiration of the people who develop these techniques. And believe me that pyro sequencing is not much used now we have got more and more newer methods of deep sequencing. And but still I want to take you through the journey of this particular pyrosequencing. So here the idea is that what about for if you want to do uh, sequencing of very large DNA fragments, let's say genomic library and all. So I'll come to genomic library sequencing, etc. But here also the idea is that let's say you have 100 kb region. Okay, then let's say if you have to go through Sanger sequencing or even pyro sequencing, at a time you will get only 5 to 700 base. So will you wait for it to end 700 yahan se mila? Maybe you start parallel sequencing from the other side. So at one time you will get only 1.4 kb sequence. Then you will wait for the company to send it, which is almost in a week's time. Then you will get the sequence. Then you will design primers. You will send to the company, which will again take 7 days to make the new primers. Then you will send it to the sequencing company कि अब तुम आगे के 700 और sequence करो, and then so on and on. So obviously it is highly labor intensive, and rather it would be time consuming method. So then the idea came that how about we break the DNA that we want to sequence into small fragments of around 5 to 700 bases, and then parallelly sequence these on a plate. So what will happen that one DNA stretch which was let's say 100 kb or so can be parallelly sequenced even though in mean shorter stretches. It is now your job to put the sequence which has come from various wells together to deduce the whole sequence of 100 kb, or in case of genome, the whole genome. So how do you arrange the whole thing after you have sequenced the whole stretch of DNA by breaking it? We'll discuss it when I tell you about genome sequencing. But here the idea is that can we do parallel sequencing, which obviously makes more sense rather than waiting for single sequence of 5 to 700 bases. So here what they have done is you break it in smaller fragments. Then you add the adapters on both the sides. So these adapters are the oligonucleotides. So the adapter that you add on one end, let's say the 5' end, it has biotin moiety attached to it. Okay. So what we have done is that in these random fragments, the ends of which we have no idea, so we cannot design primers as such, right? Because we have absolutely no idea what are the ends here. So how obviously you can design the primers already. So what I do is I add some known sequence here. Only thing is to one end, the sequence that I'm adding also has a biotin attached to it. Now, if you remember my previous class, I told you that biotin and streptavidin, they have extremely strong binding, very strong binding. So what I do is that I use beads on which streptavidin is there. Okay. So I have beads in which streptavidin is already bound to it. And when I incubate my these DNA fragments with these beads, means these DNA fragments in which the adapter is attached, just because they had biotin on this end, they can attach to these beads. Only precaution I have to take is the fact that I should use my beads in very high ratio as compared to the DNA fragments, only because I want that my beads should have one DNA fragment bound to it. Okay? So you have multiple beads. Each bead will have one DNA fragment bound to it. How it could bind? Because of the presence of the adapter, which had biotin, and we utilized streptavidin and biotin strong binding. Now, on the other hand of the this DNA stretch, we add another adapter. Okay, why do we add another adapter? Is because now we want to perform PCR. How we can perform PCR? Could we have performed PCR on these DNA fragments? No, because we didn't know the identity of any of the DNA fragment. So what I do is I add the one nucleotide, one adapter here and one adapter here, and I know the sequence of each of these. So now it means that I can perform PCR. Only thing is that I want to perform PCR in an isolated space. So I want to ensure, so I make a emulsion in a oil water emulsion. I means embed my beads such that each drop has one bead, let's say one fragment bound to one bead in it. Now, what does this oil emulsion has? So let's say I had one reaction tube in which I had all the components which are required for PCR as well as these beads, and then I sonicated to generate emulsion. And because the reaction mixture, so what I want to say that each oil droplet has all the components to perform PCR. Okay. So what is happening now is that PCR for means one particular DNA fragment has is amplified in multiple folds with the help of PCR, so that we get a very strong signal. Now, coming to let's say this is your oil emulsion, this is your bead to which your DNA fragment has bound. Now you perform PCR. So what will happen that this DNA means because all the components are there, your this DNA will make more copies. Then when you go for denaturation, it will means it will be separated, and one of the amplified fragment then can get attached to some other streptavidin moiety here. So as a result, as amplification takes place, my whole bead is now decorated with the amplified PCR products within the oil emulsion. Okay, so this is called as clonal amplification for particular stretch of DNA. And this particular thing, because you can also consider it as a microreactor. Once I'm done with the PCR reaction, I break open these emulsions. And now for individual bead, I want to perform pyrosequencing. Pyrosequencing method I've already told you. I'll not again repeat it. It is that you add individual nucleotides and so on, right? So then what I do is that I want to perform this pyrosequencing. Now you imagine that actually, though here I've shown you one particular bead, but in this tube, ideally these multiple beads are present on each of which these uh, DNA fragments have been amplified. Okay. But if I do pyrosequencing simultaneously in this tube, obviously I'll not get to know which signal is coming from which bead, and everything will be haywire. So what I do is that I pass this particular amplified DNA bead through a multi-well plate. And I smartly have made the diameter of the wells in this multi-well plate in such a way that only one bead can be entered here. Okay, so because I want that from each well individual signal should come. तो एक well में एक bead ही आएगा, ठीक है? And in each well I have these multiple, let's say, small, I don't know what term do they want to give it, but let's say multiple small beads also. And each of these beads, they are, means, let's say it has all the reaction mixtures for pyrosequencing to take place. क्योंकि अब तुम्हें इस बीट का you want to perform pyrosequencing. So what all things would be present in that well in multiple micro beads would be these, luciferin would be there, luciferase. So these are small micro beads. So ये तुम्हारा main bead है, और ये जो small orange है, these are small orange micro beads which are present there. So what do they have? They have sulfurylase enzyme, they have luciferase enzyme, and then you pass this luciferin and other things which are required for this pyrosequencing reaction. So now what will happen that, oh, okay, this is my last slide. So let me try to explain. So you see it from this particular point of view कि तुम्हारे हर बीड में let's say you perform pyrosequencing और यहाँ पे तुम्हारा first base जो था वो A था, ठीक है, and you added DATP to this whole plate. So what will happen, जहाँ जहाँ पे first base T था, मतलब उसके complementary A है, there you will see the signal, right? So obviously you will not see it in all the wells because in all wells the DNA fragments are different. So only in those wells where the first base had to be A, only there you will see the signal. So that your machine, it will detect it, and it will give you one particular signal page. Here, let's say this is a signal image where you can see, individual fluorescence coming from individual wells. It will not be there in all the wells. Then what you do next, you add DTTP. So then you will get signal in some wells, but not in other wells. So then you will get one particular signal pattern. Then you add third nucleotide, let's say DCTP. Then you will again get signal in some wells, some in not. In some you will get signal, in some you will not get the signal, right? Then what you do is that जो तुम्हारे multiple signals आए, after adding each nucleotide, you superimpose these in the machine. Means that is automatically done. So then what you come to know that okay, तुम सिर्फ किसी एक वेल को इमेजिन करो, ठीक है? तो इस वेल से पहले ए का सिग्नल आया, फिर टी सी का नहीं आया, फिर जी का आया, फिर जी का आया, फिर ए का आया. So then you come to know that okay, the sequence here is A double G A, right? And similarly for this, it will be completely, it might be different. It can be maybe the first base is A, but second base is also A, third base is also A. इसी तरह से इसका भी आया. So what I'm saying that the different signals which are perceived after each addition of the DATP, nucleotide, you superimpose, and that's how you finally build up the whole sequence coming from a particular well. Okay, so what you will have in hand is parallel sequencing of multiple DNA stretches which are present in individual wells. Okay, so this is how it is done. I do have, okay, I think I don't have in this computer, neither, okay, but means a beautiful fluorescent image comes. I will pass on the notes for massive parallel pyrosequencing to you because in TA Brown, it is just given in a small para. and you can understand it is a little complicated technique. So I've made notes for you and I will pass on those to you to make it more easier for you. But if you have some questions for now, you can please ask it. Okay, so I assume that even MPSS is quite clear to you.
Yes, ma'am.
Okay, and you're tired also because it's 10. Okay, so if there are no more questions, then in my next class, we'll be discussing the methods for genome sequencing, in which some of these techniques are used, which I've already told you. But how do you actually align the genome sequence together after doing parallel sequencing, etc., will be something that I'll be discussing. Okay, guys. Bye bye.
Thank you, ma'am.
Yeah, take care.
Thank you, ma'am.